Hi everyone, it's Dr. LeBlanc with Unit 13C, Alterations in Chromosome Number and Structure. Our learning goals are to understand some of the different types of chromosomal rearrangements that can occur in eukaryotes, to understand how gene duplications can lead to the evolution of gene families, to understand the cellular events that lead to aneuploidy, to understand how aneuploidy can affect phenotypes, including trisomy and sex-linked conditions, to understand how different states of aneuploidy can affect the phenotypes of plants and animals. Here's a typical karyotype from a human. It is a female. Here are the two X chromosomes. In a karyotype, the chromosomes are ordered from chromosome number 1, which is the largest, to chromosome number 22, which is the smallest. Chromosomes have particular names given to the regions of the chromosome. The short arm of the chromosome is often called P from the French petite, and the long arm of the chromosome is often called Q because Q comes after P. Chromosomes are also given designations based on the placement of the centromeres. Here is a metacentric chromosome where the centromere is directly in the middle. Submetacentric, where you have a shorter P arm and longer Q arms. Acrocentric, where the centromere is closer to the tip of the P arm, and telocentric, where you can barely detect a P arm at all. Mutations can alter chromosome structure. Deficiencies or deletions, duplications, inversions, and translocations can all alter the structure of the chromosomes. Mutations can also alter chromosome number. Aneuploidy is a condition where you have different numbers of chromosomes than the normal diploid number, such as monosomy, trisomy, or polyploidy. Simple translocations can occur when a chunk of one chromosome is attached to another chromosome. So here we have a chromosome translocation between human chromosome 1. We've taken this piece off here, this 2, 3, 4 piece, and added it to human chromosome 21. Reciprocal translocations occur when two pieces of chromosomes are exchanged. Of course, these are not homologous chromosomes, so it's not normal recombination that occurs during meiosis, but instead we've got different parts of different chromosomes attached to each other. Deletions can also occur. Here we're deleting this region number 2 here. We're missing it on this chromosome. And duplications can occur as well. We have this whole region with this 2-3 region here repeated here. And inversions can happen where you flip a chunk of one arm of the chromosome out and flip it around and put it back in again. So here we read the regions of the chromosome on this long arm: 1, 2, 3, 4, and here it's 1, 3, 2, 4. Certain times when you have a small deletion, such as this deletion of the tip of the P arm of chromosome 5, in a heterozygous condition, you can end up with specific phenotypes, such as this Cree-Duchat syndrome. Duplications are caused by abnormal events during recombination. Usually what happens is one of the chromosomes, when you're lining up your homologous pair, ends up with a deletion, and the other one ends up with an extra piece of the other chromosome. So here we have our duplicated region here from what both of these parts of the same chromatid are from the same homologous chromosome. And there's also a reciprocal deletion. Usually chromosomes with a deletion are lost, and chromosomes with a duplication might be carried on through into the next generation. Duplications are interesting because they can provide additional genes, the raw materials for evolution, and can form gene families. So if you have one gene and it gets duplicated, as long as one of the two genes continues to carry out the function of the original gene, the other gene is free to accumulate mutations and perhaps provide additional functionality. Over the course of many generations, the two genes may differ due to the gradual accumulation of DNA mutations. They're called paralogs or homologous genes. Here's an example of the evolution of a gene family from the hemoglobin family of proteins. So if we started out with an ancestral globin that got duplicated, the myoglobin genes evolved because they had a very particular function. They were better at binding and storing oxygen in muscle cells. And then the hemoglobins evolved their own very long multi-membered gene family. The hemoglobins were better at binding and transporting oxygen via red blood cells. And that led to the formation of these families of hemoglobins, the alpha chains and the beta chains. They have versions that are expressed early in embryonic life, and then later down the genetic locus, we have um, uh, versions of the beta chains that are expressed during the second and third trimesters of embryonic development. And then finally, we have versions of the beta chains that are expressed after birth and into adulthood. Inversions can happen either with the centromere involved, a piece of the chromosome containing the centromere can excise and flip around and get put back in again, or it can involve just one of the arms of the chromosome, a paracentric inversion. In rare cases, inversions can actually alter the phenotype of an individual if you encounter a breakpoint effect, where the breakpoint actually occurs within a vital gene, or you can have a position effect where a gene is repositioned in a way that alters its expression, say because it gets placed near heterochromatin or perhaps near an enhancer that allows this protein now to be expressed in a different tissue at a different time or a different amount. Interestingly, about 2% of the human population carries inversions that are detectable with a light microscope. So those are fairly large inversions. Most of these individuals that carry these inversions are actually phenotypically normal. However, a few can produce offspring with genetic abnormalities. Translocations can be reciprocal or often called balanced. They're caused by chromosomal breakage and DNA repair or abnormal crossovers, and they can cause position effect. Unbalanced translocations are essentially a duplication of genetic material. You break a piece of one chromosome off and attach it to another. Can also cause position effects, and they're often associated with phenotypic abnormalities or even lethalities. When you have chromosome breakage and DNA repair, you can have the repair accomplished by non-homologous end joining between two different chromosomes is going to lead to a translocation. So here we have a picture of chromosome 22 and chromosome 2. We have a homologous pair of chromosomes for each of these two chromosomes. If there's a breakage between both of them, non-homologous end joining might end up repairing these two breaks in the wrong way, so that we get a piece of chromosome 22 attached to chromosome 2 and vice versa. Non-homologous crossing over can also lead to reciprocal translocations, where during meiosis, the wrong sister chromatids pair up and you get breakage and exchange. Familial Down syndrome is an example of a Robertsonian translocation. A Robertsonian translocation is the structure that's produced when two telomeric centromeres, telomere chromosomes, are fused at their short arms. So here we have chromosome 14, and you notice it has a very short P arm and a long Q arm, and the same for chromosome 21, a very short P arm and a long Q arm. If there's a crossover between them, you can end up with this chromosome that has the longest parts of both these chromosomes, and then another chromosome that has the tiny little P arms for each chromosome. This one is usually lost. This one can get passed down. So here's a karyotype of somebody with just such a translocation. They have one normal chromosome 14 and one chromosome 14 with an extra part of chromosome 21. Chromosome 21 is usually is the chromosome that's involved in Down syndrome when you have 3 copies of chromosome 21. So essentially, this individual has 1, 2, 3 copies of chromosome 21. Here's a little girl with Down syndrome learning how to tell time. So variations in chromosome number are called aneuploidy when there's a variation in one particular chromosome or a few chromosomes within the entire set. It's regarded as abnormal. Euploidy, on the other hand, is when you have a variation in the number of the complete sets of chromosomes. It occurs occasionally in animals, but frequently in plants, especially in domesticated plants. So what are some of the mechanisms that can produce variation in chromosome number? Well, there are 3 natural mechanisms: meiotic disjunction, mitotic abnormalities, and interspecies crosses, known as alloploids. We're going to just discuss this first one, meiotic nondisjunction. So, meiotic nondisjunction can occur either in meiosis 1 or meiosis 2 and has consequences on how the gametes turn out. So what happens if you end up with nondisjunction in meiosis 1? Well, nondisjunction means that instead of the chromosomes separating from each other, they don't. So here we have the purple chromosome. It's a pair of homologous chromosomes. Each chromosome has 2 sister chromatids, and one of those chromosomes is going to go to one pole, and the other one is going to go to the other. Here in this part of the diagram, it shows that these 2 blue chromosomes, however, are both going to go to one pole together instead of separating as they should. These leads to 2 different cells, one that's completely lacking the blue chromosome and one that has 2 copies of the blue chromosome. At meiosis 2, the sister chromatids come apart, and you end up with 2 cells with an extra copy of the blue chromosome and then 2 cells which are missing the blue chromosome. So 100% of the gametes are abnormal. These gametes can produce a trisomic zygote. When it gets fertilized by a sperm or an egg joining it, it will also have just 1 copy of the blue chromosome, and you'll end up with 3 copies of the cell. These gametes can produce a monosomic zygote, which monosomy is usually not a viable condition. If you have meiotic nondisjunction in meiosis 2, you can end up with 2 normal gametes, and then the other 2 gametes will be abnormal. Here you have 1 chromosome going entirely to one of the poles, and the other chromosome, the 2 sister chromatids split apart, so you have 1 chromosome going to each of the 2 opposite poles. This diagram indicates that normally you have 2 copies of each chromosome, but sometimes you might end up with 3 copies of one chromosome or 1 copy of a chromosome. Usually monosomic individuals don't survive, and it's actually quite rare occurrence for individuals to survive with 3 copies of a chromosome. That condition is called aneuploidy, which is a variation in the number of a particular chromosome within a set. So here it's chromosome number 2 that is aneuploid. This chromosome is normal, this chromosome is normal, but this chromosome is aneuploid as well here. What about trisomies? Well, there are several different conditions where individuals with a human trisomy can survive. Here's a website that you can go to look at for individual who has trisomy 13 and another individual with trisomy 18. These individuals end up with a variety of phenotypes, but a lot of research is needed to be done in order to figure out the best way to help individuals who have those two trisomies. Trisomy 21, or Down syndrome, is the most common trisomy found in humans. A lot of research and both scientific and clinical research has gone into this state in the past 30 years, and we have been able to increase the life expectancy for individuals with Down syndrome from 25 years of age in 1983 to 60 years of age today. That's a huge leap in just a very few short years, to increase virtually more than double a life expectancy for individuals with Down syndrome. There are many, many clinical prejudices that exist for Down syndrome. Medical schools are not teaching all of the right facts about people who have Down syndrome, and there are many incorrect prejudices that are still out there. If you'd like to learn more about it, you can go to the National Down Syndrome Congress here and find out some more facts. There are many different websites devoted to people who have Down syndrome. There's World Down Syndrome Day, the Massachusetts Down Syndrome Congress, the National Down Syndrome Congress, LuMind Research for Down Syndrome. The bottom line is that people with Down syndrome live very happy and productive lives, and there's lots of evidence that you can find out there to support this. Tim Harris, for example, is an individual with Down syndrome. He owns his own restaurant, and he is also a motivational speaker. And if you'd like to have your morale boosted, you can watch his talk. It's on YouTube. And Ollie and Cameron are my favorite twins with Down syndrome, and they have a Facebook page, and they have a very inspirational video as well that their parents have put out. Euploidy is the variation in number of a complete set of chromosomes. Plants are very good at producing euploid members of their species. Bees are also euploid, and female bees are diploid, but male bees are monoploid. They have one set of all the chromosomes, where female bees have two sets of all the chromosomes. They're diploid. And individual tissues sometimes can be euploid, such as the polytene chromosomes of insects. So cultivated plants are often euploid. Cultivated wheat that you eat every time you have a slice of bread is a hexaploid species. It has 6 complete sets of all the chromosomes in the species of wheat. If you go to the garden center this spring and pick up some petunias, you might look for the ones with the biggest flowers. Those flowers are from plants that are tetraploid, perhaps, as opposed to the smaller ones that are from diploid plants. Polytene chromosomes are found in the salivary glands of Drosophila and melanogaster. They are chromosomes where several copies are lined up along from the tip of one arm to the tip of the other arm of the chromosome. They are produced by repeated replication, and having all of those strands of DNA that have been replicated remain together in one chromosome. They're all attached to each other at the chromocenter near its centromere. Each polytene arm is composed of hundreds of chromosomes aligned side by side. Here's an actual picture of a polytene chromosome from a Drosophila salivary gland stained with a dye that picks up chromatin structure. So you can see distinct bands here. Now there are also these regions that look puffy. They're called chromosome puffs, and they are active regions of the chromosome that are producing messenger RNAs that will be translated into protein. So this is what it looks like if you were to draw a schematic diagram of the chromosomes all lined up next to each other. And a puff indicates that a region of the chromosomes are looped out, presumably to have access to RNA polymerase and be transcribed into messenger RNA. So in summary, chromosomal rearrangements of eukaryotic chromosomes include deletions, insertions, duplications, inversions, and translocations. Gene duplications can lead to the evolution of gene families, such as the hemoglobin genes. While one gene fulfills the original role, the other is free to evolve new functions. Aneuploidy is a condition where an individual chromosome is present in greater than or less than 2 copies in a diploid individual. Nondisjunction in meiosis can lead to aneuploid gametes. If chromosomes don't separate correctly at meiosis 1, all the gametes will be aneuploid. If the nondisjunction occurs during meiosis 2, 50% of the gametes will be aneuploid. Human trisomies can have varying degrees of phenotypic changes associated with them. A lot of research needs to be done. Trisomy 13 and 18 individuals have severe disabilities. Individuals with very few other trisomies survive to birth. In general, most people with trisomy 21 or Down syndrome live productive, happy lives. Many domestic plants are actually euploid, having many more than 2 complete sets of chromosomes. This causes the plants to be bigger and have bigger fruit, flowers, and grains. Polytene chromosomes of Drosophila salivary glands are an unusual circumstance of euploidy. They have been very beneficial for genetic research.